So this is a re-recording of the DiFonzo seminar series that I gave on Friday, November 21st, 2025 to the biology department and associated students and faculty who were in attendance. It didn't record when we tried to record it, so I thought I'd re-record it right now to sort of capture this for anybody who wasn't able to make that event. So as I indicated that day, this was largely, let me get my laser pointer here. This was largely my presentation, but it was based on research that was done by a student, Tim Silverman, who was taking my class. So I'll tell a little bit more about that later. The title of the talk was, again, From the Lab to the Locker Room, Exploring Sleep as Biochemistry's MVP. So a little bit on the agenda for this talk. I'm going to talk a little bit about soapboxes that I sort of live and die on, if you will. Give a little bit of sleep introduction and some terminology for things that we're going to be discussing. Talk a little bit about Tim, specifically who he is as a student and an athlete and all of that, and then describe the methods, the results, and discussion based on his research project. Provide a little glimpse into what I see as future directions for this product with, again, a return to my inherent soapboxes. And then I only had the talk last about 40 minutes because I wanted to make sure that there was ample time for questions, thinking that this was likely to be a topic that would have a lot of student interest. So when we think about the soapboxes that I think a lot about in terms of the courses that I teach, one of them is sleep. So whether you have children, you remember being a child yourself, you just put yourself in the car, doesn't matter how long you are in the car, you just happen to fall asleep. And that happens all the time. I would notice it with my kids. It didn't matter how often we were in the car, they were just constantly falling asleep in the car. Turn my head around and it didn't matter what age they are, they were asleep. This is going to have relevance in a little bit when I'll talk about the fact that we can't avoid sort of that sleep pressure, that feeling, that urge to sleep. And if you don't like looking at sleeping humans, don't worry, you can look at my pets. You can look at the cats who always seem to find each other and fall asleep no matter where they were, what they were doing. You want to see cats with humans together? That happens too in my house. You want to see dogs? That happens as well. And the point with showing you all of this is to remind you of the fact that sleep is one of those things that is very important biochemically and biologically. But if you think about why we sleep, right, that's a bigger important question, particularly when you think about what's happening or not happening when we're sleeping. What's happening is we're very vulnerable, which is not the best thing when we're thinking about trying to sort of survive as a species and thinking about survival of our genes, right? We're not eating, we're not reproducing. And so it just seems to be such a counterintuitive and counterproductive sort of biological phenomenon. Yet all organisms sleep, and most organisms, except for humans, sort of don't deny that urge to sleep. So another soapbox that I talk a lot about, and it is a thread throughout the entirety of my biochemistry curriculum, is sugar. Right? So a lot of what we did during this presentation was sort of like an audience participation. So some of that might not work as well here. But I showed a couple of pictures to sort of kind of highlight that sugar is so pervasive in our world. Had a lot of questions at the end of this seminar here, sort of talking about how we have a lot in our environment that works very counterproductive to what our biochemistry has been hardwired to do for at least probably the last 100,000 years. So another soapbox that I sort of live and die on is sugar. And as you'll see, that's going to come around full circle when I sort of wrap up this talk. So why is this all important? And whether it's thinking about sleep or sort of our exposure to sugar in our environment, we are sort of a human anomaly. And apologies to any of my anthropology colleagues when I just sort of asked Google, when were the earliest humans around? Humans as we sort of think of ourselves biochemically as what we see now today. The answer that came up was about 100,000 years ago. Well, why this is important is if we think about when our sort of modern environment changed, it was probably just about 200 years ago that we had our first electric light. It also was when we sort of had our first modern climate control, sort of for an interior environment. And the idea is that 99.98% of the time that we have biochemically been around like we are as humans today, we have used the environment to provide cues, and importantly, we have not altered that environment. So what does that mean? When we think about the exposure to light we have, pretty much the only light that we had was sunlight, and that had a very specific timing. We're going to talk about in a little bit sort of our circadian rhythm and our circadian clock. You're going to notice that a lot of the T's that we're going to see in this talk are going to be highlighted and bolded in red, and that's going to come again around full circle at the end. So big environmental cue was light exposure, and that was very tightly regulated and consistent in our environment for thousands of years. And really only the past 200 years have we artificially introduced lighting at a time where we really didn't have lighting. Same is sort of true for temperature, right? And I'm going to elaborate on that point in just a little bit, but really it's only been in the last 200 years that we've been able to sort of climate control our environment from a temperature standpoint. So what are some quick take-home messages from how we think about how we are an anomaly, right? We are the only species in the world that intentionally avoids sort of that pressure to sleep. I showed those pictures of humans and animals both sort of giving in to that sleep pressure, if you will. But we are also the only species that intentionally avoids that signal and intentionally avoids it by taking things that suppress that urge to sleep. So if we think about using the environment to provide cues for us, and in a simple way, what does that mean? Well, we should sleep when it's dark out and eat when it's light out. And importantly, too, when we're thinking about eating and what we should eat, think about eating foods that resemble what they look like in nature. If it's not a food that looks like how you'd find it in nature, it's probably not something you should eat too frequently. Okay, so now on to a little bit of sleep vocabulary, if you will, some terminology that I want to make sure that everybody's familiar with because it's something that's going to be relevant to things that we're going to study in this research project. So as a way to highlight a couple of resources, if you find this really fascinating, you want to take a deeper dive, this is a great book that was put out by Matthew Walker about why we sleep. He has a great TED Talk, over almost 25 million views on this TED Talk about sleep being your superpower. I love TED Talks. This is one that's about the 15-minute range and a really great sort of insight into why sleep is so important. Another great resource, I think, if you're thinking about health and wellness and so forth, Peter Attia has a great podcast that's out there. He's got a great website with great resources. And actually, Matthew Walker and Peter Attia got together several years ago to sort of talk about had a 3-part podcast series on the power of sleep. And so I highly encourage you, if you're interested in taking a deeper dive on some of this, to look into these 2 resources. So this is a lot of place where I get some of my resources, and some of them are from some materials that I'll highlight where we get them online. So the first thing I want to do, because we're talking about sleep being sort of biochemistry's MVP, I want to talk about what sleep is and importantly highlight the 4 sleep stages that we have. So sleep is really divided into 4 sleep stages. There's awake, which really isn't sleep, but we're talking about awake time that you spend in your bed. And then there's light sleep, deep sleep, and REM sleep. And a typical night's sleep over a 8-hour time period, let's say, will cycle between about 3 to 5 different cycles through these 4 sleep stages. And as you'll see here, we bookend sort of our sleep with awake times, both when you lay in bed and before you fall asleep, and then as you wake up. But one of the things you'll notice is that as you transition through these, you go from awake, you first go into a state of REM sleep. And then you'll transition between REM sleep and deep sleep, always through this light sleep sort of transition. So you can see that we cycle sort of between these different sleep stages. You may have a period of time in the middle of the night where you have more of an awake phase, whether it's you need to go up to use the bathroom or you just find yourself awake a little bit. That's actually quite normal to sort of experience that. But the other thing that I kind of want to highlight here is that when you experience both deep sleep and REM sleep, even though you go through these sleep cycles, the first half of your sleep is actually is where you have more deep sleep than REM sleep, and it actually flips, and you have more REM sleep than deep sleep towards the latter end of your sleep cycle. So what this means, and think about this, is if for any reason you truncate your sleep schedule, whether you go to sleep later on any given night or you have to find yourself waking up early on any given morning, you are likely truncating those different kinds of sleep. So again, going to bed too late means you're going to deny yourself deep sleep. Waking up earlier than you normally do means you're going to be denying yourself REM sleep. So we're going to talk a little bit more in terms of what specifically happens during these different stages of sleep. So again, awake time, we don't think of awake time as being part of the sleep stages, but it is. Again, highlighting that before you actually fall asleep, you will be in an awake stage, but in your bed. And then before you actually physically get out of bed, you will find yourself in an awake stage again. And you also may have those brief awakenings that may happen during the night. And so for each of these slides, I'm going to also sort of highlight what is an average time in sort of a healthy sense that you might spend in these different sleep stages. You probably shouldn't be spending more than about 5% of your time in bed being awake. That's not really an efficient sleep, and we'll talk about in a little bit how we define sleep efficiency. So pretty straightforward to think about awake time. So REM sleep, again, as you find yourself drifting off to sleep, the first stage you'll find yourself in is REM sleep. And people are familiar with REM sleep. That R stands for that rapid eye movement. And here's some things that happen during that stage. Respiration increases, your heart rate will increase, temperature regulation is switched off. You may have vivid dreams, but it's very important that your body immobilizes movement so that you don't act out those dreams. But importantly, and this is super important for a student and an academic standpoint, there's a lot of memory consolidation, sort of learning, problem solving, things that happen during sort of that REM stage of sleep. And on a healthy sort of standpoint, you might spend about 20 to 25% of your time in REM sleep. So as I sort of try to compartmentalize this, I try to think about half of your sleep, we're going to get to this in a minute, might be light sleep. But if I can get about 25% of my sleep in REM, 25% in deep, that's been a good night's sleep for me. So again, REM sleep about 20 to 25%, again, dominating that latter half of your night's sleep. All right, light sleep. Again, remember, light sleep represents that time where we're transitioning between REM sleep and deep sleep. So during light sleep, your muscles relax. You may sort of have some of those twitches. Respiration begins to slow. Your heart rate begins to decrease. Again, you're transitioning into that deep stage of sleep. This is a key feature here. Your body temperature drops. We'll talk about temperature in a little bit. But to get into a stage of deep sleep, your core body temperature does need to decrease. Sleep, again, begins or transitions between cycles with this light sleep. Waking up is much easier during this time frame. Okay, and if we look at it from a sort of how much of your sleep may be light sleep, about half of your sleep could be this transitionary or light sleep stage. And then the last sleep stage is deep sleep, right? During this sleep stage, your blood pressure is going to drop. This is sort of the repair phase of your sleep cycle. You know, blood flow is going to increase to your muscles. Growth hormone is released. Tissue growth and repair occurs. Again, this talk is focused not only towards students, but student athletes. So thinking about how biochemistry and sleep can be your MVP sort of in your sport. This is a really important thing that also happens. You know, some brain things that happen in REM sleep are sort of that memory consolidation, but an important brain health thing that happens during deep sleep is your brain sort of flushes waste out of your brain there through a system called the glymphatic system. Your brain is actually going to shrink slightly when you're in deep sleep. It allows you to have fluid sort of get in there and rinse out the detritus of the day. And the inability to sort of rinse out all this detritus is going to have negative health impacts in the short term on the next day, but potentially chronically. Waking up is much more difficult in this stage. Again, you're disoriented or groggy if woken. This is an important thing to highlight too. If you find yourself trying to take a little nap during the day, right? The goal with a nap is maybe you let yourself get into REM and maybe even a little bit of light sleep, but you need to wake yourself up before you get into that deep sleep. Or you better hope that you come out the other end in a longer nap, for example, because if you try to wake yourself up from a nap once you've gotten into deep sleep, it can be really, really hard. So again, the idea with a nap is naps should really not be long enough that it allows you to get yourself into a deep sleep state. Again, if you can have your deep sleep be up to a quarter of your total sleep, that is phenomenal. One thing to highlight, though, is deep sleep is one thing that decreases as you age. So it's much harder and harder as you get older to have that very restful and reparative sleep as you get older. All right, so those are the 4 sleep stages that we find ourselves cycling between. And there's lots of ways that you can sort of measure how much sleep you have in each of these stages. So I'm going to talk about some levers now. And what do I mean by a lever? I mean something that you can actually use to push or change sort of the sleep that you have. And one of the levers I'm going to talk about, and there's a big red T there, is going to be temperature. So one of the things that we do know is that as you go through a night's sleep, your core body temperature decreases. And if you look at the scale on our y-axis here, it's not substantial. It's only about 1.5 degrees. But it is really key to get down into this lower core body temperature to promote sort of that deep restorative rest. And you probably know this, right? If it's during the middle of the summer and, it's a really warm night and you're just not able to get yourself cooled down, you know that you don't sleep really well. And a lot of people will know this. They try to just put a fan on themselves, or even in the wintertime, they may find themselves having their windows open just to have it cold in their bedroom because they just know anecdotally how much better they sleep or how much better they feel the next day when they've slept in a cold room. So we do know that. There's lots of research data that's out there on that, but there's also a lot of historical data. If we think about what early humans did, and I'm going to do a lot to reflect back as to what have we been doing over the past 100,000 years, and how is what we've been doing in the last 200 years very different from that. So humans used to sleep on the ground, and as the sun would go down, the earth would cool, and as we'd sleep on the ground, it would help cool our body, lower that core body temperature, and allow us to sleep much more soundly. So a lot of history has already been out there to indicate that our body sort of knew what we needed to do to get restorative and restful sleep. So that's one of our levers, is temperature. Here's another one of our levers, and again, another T word here is timing. And so if we think about how we expose ourselves to light, we can think about how that affects the timing of our sleep. So I want to come over here on the left first and highlight. I know this looks like a clock, but this is really a 24-hour clock that highlights our circadian rhythm. So really 12 hours on top and 12 hours on the bottom to represent really the 12 hours that we might see of darkness and the 12 hours of light. Now, really interesting, I'm sort of highlighting some of the questions that I know came up in our question and answer discussion, but asking the question about how productive sleep might be when you sleep someplace like near the equator where you have a pretty much constant 12 hours of sunlight, 12 hours of darkness, and there's not a lot of twilight in between. So we really are living in this true 12-12 sort of light and dark cycle, and it doesn't really change much, right, from season to season. And you contrast that to, say, the North or the South Pole, and depending on where they are in their winter versus summer, you may have 24 hours of either pure darkness or pure light. And thinking about how disruptive those situations can be, and is potentially that another reason why there was not a lot of evolutionary pressure to evolve away from the equators, because sleep could be so disruptive. Really an interesting thing to sort of think about in a question that sort of came up during our presentation. So you can see a lot of the things that sort of happen during what is supposed to be dark time, right? Right before you might fall asleep, you're going to have your highest blood pressure, your melatonin levels will rise. We're going to get to that sort of over here. And then you'll see at midnight, and then getting through some sort of substantial deep sleep. And then you're going to have your lowest core body temperature right before you sort of rise. One of the things that we're going to see here, and we're going to see this more on the next slide, that we're going to see these two opposing hormones. We're going to see melatonin as it's secreted from your pineal gland, and then we're going to see cortisol as it's secreted from your adrenal glands. And really, these are a yin and a yang, if you will, of hormones that either promote sleep or promote alertness and awakeness. So again, you can see when your cortisol's peak, you reach sort of your peak of alertness, and then you've got sort of your high levels of coordination and reaction times and your highest strength sort of during these daylight hours, and that's what you'd expect. So again, this circadian rhythm is really important. Light really directs, particularly the cycling of melatonin. And for 100,000 years, it's been very tightly controlled by environmental cues. Really, only in the last 200 years have we artificially introduced light that can significantly impact that. So I'm going to talk about that over here when we look at sort of what we see in the cycling between daylight hours and then nighttime hours. So what we can see here is I'm going to take this red line first, which is melatonin. And I'm going to actually start down here during the day when you have lots of light exposure on your suprachiasmatic nuclei, which is a location in your brain. You have suppression of melatonin production. And as soon as we eliminate that stimulus, that external environmental cue, we rapidly see an increase in our melatonin, and that is going to promote sleep. And so we're going to have high levels of melatonin during the night, and then those melatonin levels are going to fall eventually. And then again, we are going to wake. And part of what we see when those melatonin levels fall is we are going to see a cycling and an increase of our cortisol levels. So two things to sort of highlight, again, getting in the way of either environmental cues that have been around for 100,000 years or just how our environment has impacted sort of our mental health state. We can think about if you are exposing, particularly your eyes, to artificial light after the sun has gone down, you are continuing to suppress this red line, which means you're continuing to suppress your melatonin production. It eventually will rise and peak, but the problem is if it doesn't rise and peak until 4 a.m., 5 a.m., you're going to have a very hard time getting up because that melatonin is still going to be in its peak when your alarm clock is going off. Similarly to that, if we look at the cycling with our cortisol levels are supposed to cycle. Again, they're supposed to rise and peak in early morning and then decrease to allow us to get into a more restful state and get prepared for sleep. One of the things that can happen to keep your cortisol levels elevated is stress. And again, if you are under chronic stress from your environment and not in a way that is healthy, we will see elevated cortisol levels. And again, that negatively impacts your ability to fall into a good sleep cycle. So a good bit of biochemistry there on two hormones that we're going to see. I don't remember where this point sort of came up or this question came up or discussion when we sort of had this talk, but I'll just mention it here since it's related. One of the other things that happens is when you think about what also helps make you tired, it's the development of what we call sleep pressure. And sleep pressure arises from the increase of adenosine. And adenosine is, again, a byproduct of ATP hydrolysis. And so we're going to see the production of adenosine sort of increase as you go throughout the day. And that creates what we call sleep pressure, which is one of those things, in addition to increase in melatonin production, helps you fall and then stay asleep. So one of the things that caffeine does is caffeine actually is a competitive molecule for adenosine, and it sits sort of and it competes for receptors. It sort of blunts that sleep pressure effect. The problem is you can't do that indefinitely, and you've probably known this. If you are way overtired and you're still trying to stay awake with caffeine, you finally get to a point where caffeine has no effect and sleep pressure will sort of succumb. So just a couple of things to think about with those two hormones. Okay, so those are two levers that I want to spend some time thinking about. I want to think about timing and temperature. And just as a little preview, as we sort of talk about what Tim chose to look at with his research study, importantly, that's two variables. And as researchers, we never want to be investigating more than one variable at a time because then we don't know what we're really looking at. So Tim did a really good job with the design of his study, trying to minimize that timing effect. And we'll talk about how he did that when we look at his methods. But how do you actually then measure sort of how good your sleep is? And so thinking about some metrics that we have there. One of the metrics that is important to think about is resting heart rate. So here's one of the things that we do see that happens with your resting heart rate. So again, this is how many beats per minute. That's how we measure resting heart rate. And what we see is what we call sort of this hammock. And what happens is as you go to sleep, your resting heart rate should lower until you reach what we call a nadir or a low point. And the earlier this happens in your sleep cycle, the more rested you are. Again, if this low point doesn't happen until just a couple of hours before you wake up, you're going to find or feel probably that you don't feel as rested. And so we call this the hammock because it really should have at least an equal sort of shape on both sides. But the later in your sleep cycle, you sort of have this low point. The more it means that your body is not rested. And again, we're going to talk about how we can measure this and ways that we can measure your heart rate and look at when does it reach sort of its lowest. So that's one metric that we can look at. Another metric that we can look at, I'm going to spend a good bit of time on this because it is one of those very important metrics to look at, is heart rate variability. So when you just sort of look at a typical ECG, right, and this is sort of looking at the beating of your heart, if you will, right, we can look at, and if we looked at our heart rate, a heart rate is going to be measuring a number of beats per minute. So it's basically counting how many times your heart beats over a minute time frame. But what it's not really looking at is what is the distance of time between those beats. And that's what heart rate variability is. And so one of the things that, and this was sort of our audience participation standpoint, I was really impressed to find how many people really did know about heart rate variability. But if we sort of show these two pictures, one of them has a heart rate variability that shows a very regimented beating of your heart, exactly 1 second between each of those heartbeats. You might have both of these ECGs showing 60 beats per minute. But this heart is beating so that it's exactly 1 second between each beat, almost like it's this perfect metronome. And then we have this one down here, which has a lot of variability in the amount of time between this. And I threw this question out and I said, how many of you feel like you want to have a heart that beats kind of like a metronome and it's just like perfectly in step? And how many of you want to have this one that has a lot of variation between the time between beats? And I was really impressed to see that most people picked that they wanted this guy. They wanted high heart rate variability because that indicates you are in a relaxed state. You're going to have increased recovery if this is what your heart rate looks like, your heart rate variability looks like when you're sleeping. This indicates low heart rate variability. It's really a fight or flight state. You are going to have poor recovery if this is what your heart rate variability looks like during sleep. And again, it's not to say that this isn't an important sort of space for your heart to be in at certain times. But again, to think about it, we need to do a little bit more of a deeper dive into the science sort of behind heart rate variability. So again, heart rate variability is that variation in those time intervals between beats. And again, what it really represents is the balance between these two arms, if you will, of your autonomic nervous system, right? We know these as the parasympathetic nervous system and then the sympathetic nervous system. So if you have a high heart rate variability, you are going to be dominated by your parasympathetic nervous system, right? We know this as the rest and digest arm of your autonomic nervous system. This is when you're promoting recovery. And as it relates to sleep, this is when you are going to have quality sleep. A low heart rate variability indicates a dominance with your sympathetic nervous system. And again, that's associated with a fight or flight response, high levels of stress, overtraining, or illness. So again, this is not to say that there aren't times that your body needs to be in that sympathetic nervous state. That's not to say that it's a bad thing. The key here and the super important thing, and again, bringing in that idea that we sort of work against sort of what our environment is trying to sort of tell us, or how we are responding to the environment that we live in, is if you find yourself in a chronic stressed state, you may find yourself in a sympathetic dominant sort of state. And if that's happening when you're sleeping, you're not getting restful sleep. So if we think about what should your average heart rate be, or heart rate variability be, and I said I was really glad when I had this slide and I was able to look at it and say, because when I see students, all of my students in my biochemistry course, they all have access to Oura rings. And so when I see their data and I see that all of them have very high heart rate variabilities, I remind myself that it is an age-dependent sort of variable. And when I put myself into my category down here, I should have a significantly lowered heart rate variability compared to the 20-year-old population that are my students. And there's actually a gender difference as well. And you can see that it sort of inverts. Early in age, men actually have higher heart rate variability than women. And then it gets much more similar and even inverts as women have higher heart rate variability as they get up into their 70s. So an important thing to highlight with heart rate variability, again, not only is it highly sensitive, can vary from person to person, change daily or even hourly, it really is an important metric that you look at from a relative standpoint, meaning that you compare relative to changes that you make in your own body and environment, right? Relative to your personal baseline. As much as you might compare it to somebody else and just see how it compares, you really don't want to be doing that comparison, because the best comparison is with metrics or levers that you can change yourself. So again, factors that you can't fully control, obviously age and genetics, hormonal changes are probably beyond your control. You can't control whether you're ill or you're inflamed, for example, after some exercise stress. Environmental factors may or may not be out of your control. I'm going to say that stress and environmental factors, maybe they are out of your control, but the goal with what you learn here today is recognizing what ones are within your control. So again, the sleep quality and duration is a lever that I hope I convince you today that you can control. You can control whether you're taking caffeine or other stimulants that may negatively impact sort of your sleep cycle. Alcohol and other medications can negatively impact things. Physical activity, we'll talk more about this when we talk about the specific variables that Tim looked at in his study. But thinking about ways that you can maybe control your stress a little bit better, maybe doing some mindfulness or meditative breathing, thinking about doing things to better take care of your mental and emotional state. And then again, bringing in that other soapbox of sugar, right? Thinking about nutrition and hydration. All right, so what are the ways that we can measure how well we sleep, right? All of us want to get sort of a gold star or a gold medal, to sleep well every night. But nobody wants to be hooked up to, some cumbersome machine that measures all of these different metrics. It would be great if we had a simple way that allowed us to sleep in our own bed, yet still measure and assess how well we sleep, and then try pulling certain levers to see if we can make changes and see how that improves. So the Oura Ring is what I have used. I've used it personally for like 6-7 years to sort of monitor sleep. This is what the Oura Ring looks like, right? It's just a ring that you wear. It's got several biometric sort of sensors that are in there. And again, these are research grade sensors. They adapt to your unique physiology. They collect and deliver accurate continuous data day and night. So basically, if you have an Oura Ring, you may notice from time to time you see red lights or green lights that are sort of underneath it. Those red and infrared LEDs are going to be what are measuring your blood oxygen levels while you sleep. Green and infrared LEDs are going to alternate to measure your heart rate variability, and your respiration rate. There's also negative temperature control or coefficient sensors that track your temperature trends. And then there's an accelerometer that tracks movement and activity 24/7. So what's really fascinating about all of this is how accurate they are to sort of the gold standards for how we measure these things. So compared to ECGs, there's a 99% accuracy for heart rate and a 98% accuracy for heart rate variability. Lots of women's health metrics are also measured. So this is their ability to predict ovulation. And then even though sleep tracking is not as high, it's still pretty darn good when sort of compared to clinical polysomnography, which is sort of the gold standard for sort of measuring and tracking sleep. So one of the things that's neat that the Oura Ring can provide for you is a sleep score, which is basically a score from 0 to 100 that tells you at a glance sort of the quality of your sleep. And then you can think about pulling certain levers and see how your sleep score is affected. So basically, a B grade or higher, 85 or higher if you will, your sleep is optimal, keep it up. You'll get a little crown there to indicate that you're doing well. 70 to 84, your sleep is okay, you're on the right track, but certainly there are areas that you could improve. And under 70, something is off with your sleep. Balance might be off, maybe some lifestyle changes that you can make to improve it. And these are things, again, that you can watch, and Oura is intuitive to, and it'll ask you when you get up the next day, hey, you okay? Didn't seem like you slept as well last night. So how it calculates this sleep score is a combination of several metrics, and I'm going to take a minute and describe how these are calculated. Some of them are pretty straightforward, right? When you think about total sleep, it's basically just saying, did you get enough sleep? Well, what is your efficiency? We're going to talk about that more on the next slide here, but how much of your time that you spent in bed did you actually spend sleeping? Remember, of those 4 sleep stages, we have awake, and then we've got REM, light, and deep sleep. If all 4 of those represent our denominator and we take awake out of the picture, all of those other 3 stages represent our numerator. And again, the closer you are to 100, then the more efficient your sleep is. Remember, we've got that ability to sort of tell how movement is happening during your sleep. So how restful was your sleep? And again, through different metrics, it's able to monitor pretty accurately whether you're in REM sleep or deep sleep. Latency is something that measures how quickly you fell asleep. Do you remember when we showed that graph that showed cycling between awake and then REM and then light and then deep? You kind of like go down that roller coaster, if you will, and as you're descending into deep sleep, how quickly you go from awake to REM sleep determines your sleep latency. If you are falling asleep too quickly, you might say, hey, that's super great, I'm pretty efficient. But actually that indicates poor sleep latency, which probably indicates you're overtired. And timing, again, is super important. When are you actually getting to sleep? Are you going to bed according to what your natural circadian rhythm is? And one of the things that we can note is as you alter sort of your sleep cycle, plus or minus more than about an hour, that can indicate and have some problems with what we call social jet lag. And we'll get to that a little bit later. So again, I highlighted already, when you calculate sleep efficiency, when you talk about the total time that you have in bed, which is monitored with all 4 of those sleep stages, and you eliminate awake and you just say how much time are you actually spent in sleep cycles, that is going to calculate your sleep efficiency as a percent. So the lever that I'm interested in sort of investigating here is temperature, right? We'll talk about timing and how Tim had an ingenious way of making sure that that was not a variable that he was measuring. But if we think about how we can control temperature, I use something called a chili pad. So a little bit of history as to how that evolved. A number of years ago, I reached out to Tara Youngblood, who is the CEO and co-founder at SleepMe, which creates the chili pad. And I just reached out and basically said, can we schedule a call? I'm interested in sort of developing a research project with college students, and can we develop a collaboration here? And Tara was very interested, and she went ahead and she sent us at the time 4 of what they had as chili cubes. So they were twin size mattress pads. This is sort of the cube portion of it. You control the temperature of the mattress pad with this, and it circulates water through the mattress pad, and that's how it controls temperature. So at the same time, I reached out, even though Oura rings have always been my metric, I wanted to use it as a metric for that study. So I reached out to Jeff Collins, who at the time was the head of business development at Oura. He's currently the director of human performance. And I basically sort of said, hey, we are doing a study. I'm collaborating with folks at Cryo Inc., which were the creators of the chili pad. and worked out, and I had a grant to actually pay for all of these rings. I have about 20 different rings, variety of sizes to fit students, and off we went. I had 20 rings, I had 4 chili pads, and we were ready to start this study. I had 4 students who were super excited to sort of take their chili pads home. We had all kinds of ways to collect and monitor our data. Again, they were ready to go. The date for this picture was January of 2020. And it was shortly thereafter that literally life and this research project was put on hold. As we talked about when I gave this talk, I wasn't even allowed to share paper with my students, much less bedding material. So we were kind of in a place where I had to table this project that I had a lot of momentum sort of going on. So then this project sort of sat, and then along comes Tim Silverman. So here's Tim. Tim is currently a senior at Mercyhurst. He's an applied forensic science major, and he is a member of the men's lightweight rowing team. So Tim is currently taking my class, my Biochem 1 class, and he's also a student in our honors program. And so he approached me and he said, hey, I'm really interested in this sleep stuff. I actually have a Garmin Forerunner watch, and now that I have the Oura Ring as well, what do you think about trying to do something investigating sleep? And here's what came out of it. This is the title of his research paper as he had it for his honors program, The Bone-Chilling Biochemistry During Sleep, an Analysis of Surface Temperature and Sleep. So I'm going to walk through now sort of the data that he gathered through this study. So his guiding research questions were, in what way does mattress surface temperature, and again, you're going to notice some of these red bolded T's here. So in what way does mattress surface temperature correspond with objective sleep quality measures, that is total sleep time, sleep efficiency, the amount of deep sleep, and the amount of REM sleep? Is a cooler sleeping surface, that is like 55 degrees, conducive, that should say to enhanced restorative sleep when compared to a neutral or a warm temperature or not controlling temperature at all? And in what way does mattress surface temperature correspond with subjective sleep quality? So those are his guiding research questions, and use those to then form 3 hypotheses. First hypothesis says cooler mattress surface temperatures, such as 55 degrees, will lead to higher sleep efficiency and or longer periods of deep sleep when compared to warmer 74-degree conditions or control conditions. Warm mattress temperatures, such as 74 degrees, will be linked to more awakenings, shorter sleep time, and lower heart rate variability values. And then third hypothesis was that subjective ratings of sleep quality will match objective improvements in deep sleep and heart rate variability during cooler temperature nights. So those were his guiding research questions and his hypotheses, and here's how he actually investigated that. So his methods. So how did Tim actually sort of measure all of this? What were his methods? So he developed what we call sleep blocks. So these were 10 randomized blocks of 4 individual nights where there was a night spent under a control sort of situation where the temperature controlled mattress pad was turned off. He had a block where it was set to a cold temperature. He had a block where it was set to sort of a neutral temperature, and then a block where it was set to a warm temperature. So when I say that these blocks were randomized, he basically preset up a matrix where it was a randomized sort of selection for which block would be night 1, night 2, night 3, and night 4. And he always had 4 consecutive nights. So again, trying to eliminate any variability that we have there. And he completed over the course of the semester 10 of these randomized blocks, so 40 individual sleep nights. And this was a great way to think about reducing bias, right? If you had a situation where you always knew the first night of your study window was going to be the heat was, or the temperature control was off, and then you'd have a cold night, and then you'd have a medium night, and then you'd have a hot night, that could introduce bias. That random sort of assignment of the blocks also allows you to control for external variables like stress, individual variations in schedule, maybe what your caffeine intake was. But that was a really neat way to sort of control for some variables that maybe are not controllable. And that one of those big things is sort of participant bias. Now, to that end, part of the challenge of this study was it was an N of 1. Tim was the only participant in this study, so certainly that introduces its own sort of challenges to interpreting this data. But one of the things that Tim did very, very well to try to eliminate sort of that variable of timing is he made sure to have a consistent bedtime, plus or minus 1 hour on any given night. We asked him afterwards what that was, and he said it was 10 p.m. So on any sleep study night, he was in bed anytime between 9 p.m. and 11 p.m., not any later than that. He also tried to have consistent levels of physical activity, so that wasn't necessarily a variable that would affect his sleep. And then importantly, on sleep study nights, he had no alcohol, and he also avoided large meals or heavy exercise within 4 hours of going to bed. So really, really did what we need to think about doing when we design an experiment, right? We want to make sure that we are measuring the variable we'd like to measure, which in this case was temperature, and doing everything that we can to minimize other external variables, importantly, including our own bias. Now, I told Tim at the end, I said, harder to do here with an N of 1, but one of the only ways that we could have made this sleep study be a little bit better is if he didn't even know what that sleep block assignment was going to be. Have somebody else sort of sit and program sort of this control pad for you so that you didn't even know when you woke up the next morning what it had been set at. So the sleep procedure is basically to set that assigned mattress temp 30 minutes before going to sleep. And again, I just highlighted the only way that this could have maybe been made a little bit better is if he had someone else sort of program that so he didn't know what kind of a temperature it was going to be. Now, he probably could have figured it out, right? He gets in and it's either going to feel pretty cold or it's going to feel quite warm. But that would have been another way to eliminate sort of that bias. We call that blinding a study when even the participants don't know what's going on. And again, making sure that he went to bed within plus or minus an hour of that 10 p.m. sort of set time frame. And then basically let yourself fall asleep. So obviously I don't have a picture here of Tim falling asleep here, but fall asleep and then the next morning gather your data. So here's the data that you gather. Basically from either the Oura Ring or the Garmin. The data that I'm going to show you today is largely from the Oura Ring because that is what was part of my original research study. So gather your sleep score from those devices and record what your total sleep time is, your deep sleep and your REM sleep, the number of awakenings, sleep latency, sleep efficiency, average heart rate in beats per minute, heart rate variability in milliseconds. And then Tim also gathered a subjective score. Every morning when he woke up, basically on a scale of 1 to 10, how do I feel like I slept, right? And again, the subjectivity of this score, the only thing that could make this research study better is if he didn't know what the sleep temperature was that was set the night before, whether or not that could negatively impact or in any way impact the subjectivity of his score. Things that I've got underlined here, these are going to be things that we found to be most significant or most interesting for us to study further. So here's some of his results. Oh my gosh, lots of data tables. Okay, lots of data tables that we have here, and these are super important. Lots of statistical work to do here to find whether or not things were statistically significant, but I'm just going to give you the highlights and the results in an easier to digest format. So here's what we found with regards to sleep score. So again, what you'll see in the next few slides is I'm going to show the same 4 blocks that we'd see in that sleep study block. The gray representing sort of the control where there was no temperature control. We're going to see the cold represented by the blue box, the neutral temperature represented by the green box, and then the warmer temperature represented by the red box. Anytime you see a red asterisk, that's going to indicate that that data was statistically significant from the rest of the data. So, and what you'll see as numbers within the box are what those metrics were. So remember, the sleep score is on a scale of 0 to 100. So you can see that Tim had scores here that were in the high 60s to mid 70s. Same data, but represented as in a graphical format here, where you can see sort of the error bars within each set of data. And again, that was statistically significant. So in research speak here, we can say that that 55-degree temperature produced significantly higher Oura ring sleep scores than all other temperature settings. That is the 68 degrees, the 74 degrees, and the no control or the off setting. And that was a statistical significance with a p-value less than 0.05. When we look at heart rate variability, we see something pretty similar. We can see, and again, these are the heart rate variability values. Tim has very good heart rate variability, right? I indicated that I'm sort of a typical, early 50s woman, that I am sort of in the 20s and 30s for my heart rate variability. So we can see here statistical significance in sort of the cold setting and that neutral setting. Again, we can see this looked at in a graphical way here. And in statistical speak, we can say that the 55 and 68 degree temperatures showed significant differences in heart rate variability between the control setting, again, when the mattress pad was set to off, and a statistical significance with a p-value of less than 0.05. There was no statistical difference that was observed with that warmer setting, that 74 degrees. So just flipping back here for a second and showing you that sleep score, statistically significant for the lowest temperature. And then the same thing for heart rate variability, statistically significant with both that low temperature as well as that mid-range temperature. Now, when Tim looked at his subjective ratings, again, looking at a 10-point scale and saying, how well do I feel like I slept? It was statistically significant at that lower temperature setting. So that 55 degrees produced significantly higher subjective sleep scores than all of the other temperature settings. So the 68, the 74, and then the no temperature control with a p-value less than 0.05%. So what does this all mean? And I just took a few quotes from sort of the discussion and conclusion section of Tim's paper to sort of highlight this. So the findings of this study indicate that the temperature of the mattress, there's again that red T again, the temperature of the mattress surface can have an impact on both sleep quality and recovery, and the changes can be measured even by non-lab grade wearables. And that's what's so key about this study, is it something that can be easily done in an external environment and done by college students. From a biochemical standpoint, better quality sleep means more effective recovery for processes like ATP replenishment, tissue repair, hormone regulation, all of which depend on restorative sleep stages. And then if we're thinking about athletes, students, student athletes, again, anyone dealing with limited sleep, the quick modification of temperature might be the simplest way to enhance recovery without medications or major lifestyle changes. Okay, so where do we sort of go from here, and what are some actionable things that I can sort of tell you about? Well, one of my passions is speaking as much as I can to anybody who wants to listen. And so I've talked to the news a lot about how to get good sleep. So again, whether it was talking to them about tips for a good night's sleep, talking about how caffeine can sort of have negative effects. They have frequently called on me when we have our daylight savings time changes and how those can sort of affect you physiologically and biochemically. If we think about what these tips for a good night's sleep are, we've talked about two of them throughout this entire talk, right? We've talked about the importance of the timing of sleep. So important as a variable that Tim made sure that it was not a variable in his study. He kept the timing of his sleep consistent. When you think about, and if you want something actionable from this, really keep the time that you go to sleep and wake up on any given night not different by more than an hour. And I know that sounds crazy to tell a college student, right? You get to the weekend and you're like, you just want to have sort of that social experience that you can't have during the week because you've got classes. But when you adjust your sleep time and your awake time by more than an hour, it introduces what we call social jet lag. So that really is that awful feeling that you can get when you actually have physical jet lag, right, from travel. That actually happens on the weekends when you do that adjusting of your sleep schedule by more than an hour. So think about that timing aspect. And I promise you, you will feel better if you do your best to try to not change your sleep time and awake time by more than an hour. We've obviously talked a lot in this presentation here about temperature. So doing the best that you can to control sort of your ambient room temperature, right? Not everybody might have access to a temperature controlled mattress pad, right? But doing the best that you can to make sure that you turn down temperature. So again, some of the questions that came out during our question and answer session were, it's known that Grandma and Grandpa's house always seems to be super warm because they like keeping their house warm, but maybe that really doesn't help them with sleep at night. And sleep gets more and more dysregulated as you get older. So, you save money too, turning down the thermostat at night. So timing and temperature, again, were two things that we talked a lot about in this presentation. So another T to think about is technology. And what I mean by that is think about how technology in this last 200 years is so different from the 100,000 years of biochemistry and physiology that we have evolved with, and how much that technology can impact our melatonin levels, right? By having light that's shown in our eyes and on our skin after the sun has gone down, and thinking about how technology just in and of itself can induce stress and affect those cortisol levels. So two hormones to sort of think about and how technology can negatively impact those. Now, food doesn't start with the letter T, so I just call it treats here to remind ourselves that when you are asking your body to do biochemistry and physiology after the sun has gone down based on food consumption, that can ask a lot of your body that, can impact your sleep negatively. So not only treats in terms of food, but thinking about alcohol consumption, thinking about caffeine consumption in particular, as you get to a point after the sun has gone down. And the last thing to sort of highlight here is treatments. So if you are somebody who struggles with sleep and you've gone through these other 4 T's, you've, optimized your timing, you've done your best to optimize your temperature, you've minimized your access to technology within 2 hours of going to bed. And the same thing is you're making sure that after the sun goes down, you're minimizing what food and other things get put into your body, and you're still finding yourself struggling with sleep. It might be time to seek out medical attention and find out if there's treatment options that are available. So that's your 5 T's that help with a good night's sleep. Well, I guess now I have a 6th T. It's Tim and Tim's research. So what are some future directions that we can take this project, and how does it sort of tie in with my previous soapboxes? Well, again, my other soapbox is sugar consumption, really, and that's problematic. A question that came up in our question and answer session is, if you're going to eat something before you go to bed, what is a good something to eat? And I would say probably not something that's high in sugar. But if I marry together my soapbox of sugar and my soapbox of sleep, is there a way that we can think about doing something with both of these? And in a very exciting way, one of the things that Aura has done is they've partnered with a company called Stello to look at CGMs, or continuous glucose monitors. And again, this is a way of monitoring in real time your blood glucose levels. And when we might partner that with the ability to control the quality of our sleep with a chili pad, and then we add in how can this affect our student athlete population, we might have the makings of a really neat and robust study to look at moving forward. So again, enhancing that metabolic health using this combination of a continuous glucose monitor to find out how food sort of affects your body, monitoring sort of lots of health metrics, including heart rate variability and sleep, and then trying to optimize our sleep with temperature control could be sort of a great way to fill in this soapbox, 2 soapboxes that I have, right? So some really great things that you can get from using that continuous glucose monitor that integrates with an Aura ring. So beyond that, right, we know that the Aura ring can monitor lots of metrics around sleep and rest. From a student athlete population, really monitor some great things with activity and fitness, and not necessarily a prong of this research study or what I'm proposing for future, but there are a lot of metrics that are really important to women's health that can be measured by some of the things that we're looking at here. So the goal here is that we might be able to add to Aura's already robust body of literature, right? We can even see here that they are looking at research projects in college students. So a really neat way that we might be able to supplement the body of literature that's already out there. So I made sure to highlight some important sort of books if you're interested in taking a deeper dive on any of this, right? I already mentioned Matthew Walker's book, Why We Sleep. I mentioned Peter Attia with his podcast, particularly his 3-part series with Matthew Walker on Why We Sleep. But he's got a great book here talking about the science and art of longevity. Anybody interested in going into the healthcare space, I think would find some really valuable insights in there. And then again, on this sugar soapbox, if you will, a really great book to think about how it's important to manage the cycling of your glucose levels, right? Biochemistry students know that my entire course is taught with the thread of glucose homeostasis, and sort of thinking about ways to think about balancing your blood sugar levels is super important. So at this point, I think the talk was only 40 minutes at this point, and we stopped to sort of have people address questions, right? What were things that were interesting? I tried to sprinkle those in as I thought about them as we sort of talked about things here, but I'll encourage you, if you've watched this afterwards and you're interested in this topic and you have some questions that I didn't address already, don't hesitate to reach out. Thanks for listening.